everyone, and welcome to today's episode of Seed Speaks. My name is Ashley Robinson, and I'm the editor of the Alberta Seed Guide, and today I'll be acting as your host. On today's episode of Seed Speaks, we're taking a closer look at what techniques modern plant breeders are using to bring you the crop varieties of the future. We have a few guests here today to discuss this with us. Thank you to Beni Kaufman, Ron Knox, and Tom Warkentin for joining us. And to those watching, please don't hesitate to ask any questions you may have for our guests in the chat box. And let's get started with getting to know our panelists. So, Beni, can you please tell me a bit about your work at Agriplex Genomics?
Sure, I'll be happy to. We are a genotyping company. We have our proprietary technology called Plexi, and we provide genetic markers, genotyping for animals and plants, and that's about it.
Perfect. Thank you so much. And next up, Ron, could you tell me a bit more about your work at AAFC and Swift Current?
Yes, thanks, Ashley. Yeah, I work as a research scientist in the wheat group at the Swift Current Research and Development Center in Saskatchewan. And a lot of my work revolves around testing breeding material for various traits, including disease resistance, but quality agronomy, using marker technology. And I also oversee a double haploid program in the breeding program. And also I'm involved in some traditional phenotyping work.
Thank you so much. And I'd just like to also take this moment to acknowledge that Ron won the Plant Breeding and Genetics Award this year, which is part of the Canadian Plant Breeding Innovation Awards. And our own magazine, Germination, and Seeds Canada are sponsors of it. So congrats once again, Ron.
Thank you.
And certainly, last but not least, Tom, can you tell me about your plant breeding work at the University of Saskatchewan?
Yeah, sure. Thank you, Ashley. Yeah, I'm Tom, and I'm a pulse crop breeder at the University of Saskatchewan in a group called Crop Development Center. Most of my effort is on breeding pea, but I also have a program on soybean, which is a relatively new crop in Saskatchewan. It's a huge crop in the world, but in this part of the world, it's quite small yet. So my work is on plant breeding and research related to pea, especially, and to some extent soybean.
Thank you, Tom. And okay, let's get going on questions. So my first question is for Ron. I know a lot of your research has focused on breeding for disease resistance in wheat. Can you tell me about a few of the breeding techniques you've used for that research?
Sure. One of the big areas has been the application of molecular markers, DNA markers, for helping the breeders select the best lines in their breeding programs to improve their gene pool. So we tend to try to apply the markers at the early stages of breeding, so that the breeding resources are focusing on the best material in the breeding program. We're also applying genomic selection for more complex disease resistance. Right now, we're trying to focus on Fusarium head blight in durum, and we do assessment for various facets of FHB resistance, such as incidence or severity, and resistance to the toxin, deoxynivalenol, which people will know as DON. And we also do traditional breeding work with our markers. A lot of them are correlated markers, so we sometimes, have false positives. So we really want to follow up with traditional breeding methods with disease inoculated nurseries in the field to make sure that we're selecting for resistant plants.
Thank you so much. And Tom, I know you mentioned, of course, that mainly you do a lot of pea breeding, but you've also recently gotten to soybean breeding. So can you tell me about the breeding techniques that you use in your lab?
Yeah, sure. I would say that we use conventional plant breeding methods supplemented with marker-assisted selection and other genomic tools, as well as phenotyping tools. By conventional plant breeding methods, I mean making appropriate crosses, growing early generation, nurseries, yield trials, developing breeder seed, and finally releasing varieties. And in terms of the marker-assisted selection in a crop like pea, where there's not a massive amount of work in the world on this crop, so we conduct a lot of the genetic studies that give rise to QTLs and marker trait associations, and then we try to make use of that information to develop breeder-friendly markers. As Ron said, to implement those early as possible in the program, so that we can get rid of the seeds, the plants that we don't want, and keep the best ones to go to the field. Maybe I could also say that, we're also beginning to explore genomic selection in pea and soybean, to select better, to make better crosses, and B, to select better progeny. Maybe other, you could say, modern tools that we're using in certain situations, something that people often call speed breeding. We'll use that approach for the most important crosses. Some of us in our, I'm speaking of me and also my colleagues in Crop Development Center, are using the drone technologies for rapid phenotyping, and some activities on using some rapid techniques to do root system architecture phenotyping as a tool to select better lines that might do better in terms of drought tolerance or lodging resistance.
Thank you so much, Tom. And both Tom and Ron touched on this. And of course, Beni, you do a bit different work than Tom and Ron do. But can you tell me a bit more about what exactly is molecular marker technology and why it's important to plant breeding?
Sure. So molecular markers are a tool or a group of methods that enables us to interrogate the genome and trying to understand it with higher or highest resolution. So that means at the DNA level. We heard here about different kind of selections that our colleagues use. The basis of being able to do this is knowing about the genetic variation that exists in these plants. So once you gain this understanding, you're able to associate this genetic variation to phenotypic variation that is observed, and with that, you're able to. This is the basis to the different breeding schemes or breeding programs that were mentioned. So it is used to understand the foundation and the blueprint, if you will. It also is being used to monitor the changes that you want to introduce over time for this purpose or the other. Obviously, we're focusing here on molecular breeding, but molecular markers are used in other fields of study, in evolution, ecology, conservation, medicine, and other fields.
Thanks, Beni. And of course, so far, I feel like everyone can follow along and see that you guys are kind of all related a bit in the work you do, and you all work in genomics. So can you tell me about how genome sequencing has pushed plant breeding forward? And I'll just throw that out, whoever wants to start answering first, and then you guys can kind of build off each other's answers if you'd like.
Sure, who wants to start? Maybe I'll start because I'm the technical guy. Yeah, maybe I'll start because I'm working on the foundation, and then they're doing the breeding. So as I said a minute ago, the foundation of molecular breeding is understanding the genetic variation. So genome sequencing provides the most granular resolution into this variation. Having a view of the genome at the nucleotide level is what fuels the development of not only breeding processes, but also the emergence of methods like genetic engineering or gene editing, where we are no longer just taking advantage of the variation that we have uncovered or found, but also directly affecting how it changes, or we can introduce variation actively.
And Tom or Ron, is there anything else you'd like to add?
I can
Yeah,
jump. Go ahead, Ron. Okay, I can just jump in and saying that, yeah, I think the genome sequencing activities in my own career have been quite interesting and helpful. I was part of an international consortium that sequenced the P genome. We published that in Nature Genetics in 2019 after about 6 years of hard work amongst many people. I can say that several of my plant breeding colleagues here at the Crop Development Center have been involved in similar consortia. Curtis Posniak working on wheat and other cereals, Kirsten Bett on lentil, and Bunyamin Turan on chickpeas. So within our unit, we've done quite a lot of activities related to developing these genome sequences related to the crops we work on. And I feel like, as Beni said, it's been really helpful to understand the sequences at the granular level, as you said, and in that way, from our maybe practical point of view, to help us develop better markers, more effective, more reliable markers for traits of interest. So those would be my comments.
Yeah, just to follow up on Tom, I guess for me, I've been doing this for quite a long time, and so really when markers were just conceived for use in plants, and we struggled a lot in the early days to have markers, and they were quite complex to apply and not very user friendly to the breeding program. So with the modern technologies and the number of markers that we have access to now, for somebody that's involved in molecular genetics such as myself, it's a little akin to a child in a candy store. It's a wonderful thing, but at the same time, it's a little overwhelming with the wealth of information that can be generated these days, and we have to pick and choose what we want to work with, and we have priorities. Each researcher probably has their specific pet genes that they're interested in, but it's a huge resource for us to improve the quality of the markers and to improve the precision of the markers. So we now have reference genomes, for example, in wheat, one of the most complex agricultural crops in existence. And so having a reference genome really helps us to improve on the markers that we have and expand on markers that we have. So it's really a beneficial move forward.
Thank you. And just listening to you guys talk there, that reminded me of course, Tom mentioned he was involved in the sequencing of the P genome. And then you mentioned that your colleague Curtis was involved in the wheat genome, which I think Ron was also involved in that sequencing. Am I correct? Yeah. So can you guys?
That's right. Yeah.
Yeah. So guys,
with the leadership of Curtis Posniak, sir.
Yes. So can you guys just quickly talk about why it was important to be able to have these sequences done for moving plant breeding forward? I think you guys have kind of touched on it a bit with being able to focus on those different traits and everything, but can you just maybe touch on it a bit more about what having those genome sequences meant to the world of plant breeding?
Do you mean like having the reference genomes or yeah?
Yes.
So I can tell you from my side, so our bread and butter are molecular markers. Now, to be able to reach and use or do the interrogation that I was talking about, you need to develop assays, and the having the reference genomes made it so much more efficient, so much more accurate. You cannot have a molecular marker data point or data points without having the right primers in your PCR reaction or in your sequencing reaction. And the reference genome is the tool that enable you to develop those. So this is one point. The other one is that to facilitate those selection methods that were mentioned earlier, or any marker-assisted selection program, you have to have a good idea where those markers are located. Without that, everything else will be much harder. So the reference genome gives this, if you will, topographical context to the markers, and that is really the tool that enables understanding linkage, that enables doing mapping, that enables doing, if somebody wants the physical cloning of sequences or genes and so forth. So it is very, very valuable. We would not be able to do our work efficiently without it.
Mm-hmm.
Thank you. And Tom and Ron, of course, you guys have been involved in these genome sequencing to make these reference genomes. So can you guys just talk a bit about what it meant to you guys to be able to have those now for yourselves, for your own breeding work?
Ron, you go ahead.
Okay, thanks, Tom. Yes, so in developing markers, so the type of work that I've been involved with, there's a level of kind of chance and randomness to the work in the past. We'd find what we call polymorphism in the DNA, so differences in the DNA between different individuals that express different traits, and so we'd use these variations in the DNA spotted throughout the genome, and then we'd try to correlate those with the phenotypic traits, so like disease resistance. And so that was always kind of a chance process to try and come up with a marker that was linked to the trait of interest. Now with reference genomes, the fact that we have sequences, all the sequence is laid out, and it's in a way every base in the DNA is numbered and organized. So we can now do searches on that sequence. If we have a piece of DNA that we know is related to a trait, we can do a search on that reference sequence, and we can find out where that piece of sequence associates. And then, because we have the sequence surrounding it, then we can pull out sequences and look amongst individuals that show phenotypic variation. So maybe a susceptible versus a resistant line for a disease such as Fusarium head blight. And then we can work to develop markers that are closer to that sequence. So that's on one end. The other end is geneticists do know something about the genes, and the reference sequence provides us full sequences of genes. So it allows people to look for variants of those genes, which we call alleles. So it allows us to search for improved types as well. And yeah, it just provides a wealth of tools for us.
Thank you. And Tom, is there anything else you'd like to add?
Yeah, maybe one other comment. I certainly support all the comments that were made by Beni and Ron. But in addition, maybe sort of a little bit of the human interest aspect, like in developing in these international consortium experience I had, it was led by colleagues in France. We had participants in Australia, in US, Canada, Czech Republic. And so we had a lot of meetings over a lot of years, sometimes very late at night, sometimes very early in the morning to accommodate all the time zones. And I think it led to, a good spirit of collegiality, and it's led to further collaborations in the future. We're now have sort of a second phase of that activity to develop a pan genome for pea, and a similar thing has taken place already, I believe, in wheat and other crops. So sort of the aspect of bringing people together and on common themes has been really another real benefit of these activities.
Yeah, definitely. It does bring together people, and of course that leads to future collaborations. So next, I'd like to ask a question. We had a question from the audience here, and I don't know if this, maybe to some of you, this won't be the most relevant because it's asking about tomatoes, but we have a quick question from Najit. I'm sorry if I'm pronouncing your name wrong. But what do you think about the new tomato varieties? Those are claimed to be resistant to I'm not going to try and pronounce that gene. Does molecular marker of that already clarified? Any comments on that?
I don't know the specifics. Maybe Beni can comment if you know the specifics.
Well, I'm not familiar. It looks like some sort of resistance to some virus, obviously,
Yeah.
but I'm not familiar with this particular one. But I can tell you that we have a panel of markers for tomato in Agriplex Genomics, and unless there is some complicated biology here for this virus, I think that there is a wealth of molecular markers for tomatoes. So like any other disease, especially in tomato, where there is introductions and there is high familiarity with wild relatives, it's a question of exploring and finding resistance, either species or accessions, and then our colleagues here can use methods, or obviously not there by similar tomato breeders use the schemes to introduce it. I'm sorry I cannot give more specific information for this particular pathogen or disease, but as was mentioned before, molecular markers are the tool to enable the breeding for such resistances.
I'll do a quick plug for our own magazines. I know on the Seed World and European seed sites, we have a news release or some information up about that on there. So if any of you guys here on the panel are interested, I would recommend you check out our websites after to learn a bit more about it. And my next question, of course, we've really talked a lot about genomics and all the different technology there. But of course, as researchers working in the world of plant breeding, I'm guessing you guys also keep up to date on other breeding techniques. So are there any ones that catch your attention that you'd like to share that maybe you're not working with yourself, but you're still following and are quite interested in? And I'll just let whoever feels like tackling that question first answer.
I will not be the first this time.
Well, I think Tom mentioned work with phenomics. That's something that I think will have a lot of potential going forward, and we're trying to work that into our breeding work. So phenomics and high throughput phenotyping. I think those provide opportunities to look at our plants in more detail in terms of the phenomics, looking at traits as they progress over time. So it provides an additional dimension to our understanding. And so, for example, with disease resistance, we often only have the resources to go and rate our plants one time in the field. And so we lose a lot of information that way. So with, high throughput phenotyping technology and phenomics, I think we can really increase our understanding of how pathogens interact with the host and through that improve our disease resistance in the future.
Thank you. And Tom, would you like to share something you have on your mind?
Sure. I'm surprised we haven't really focused on gene editing yet. I think Beni mentioned it a bit earlier, but so that's certainly a technology that's very interesting. It's in the news a lot, in the scientific news. It's kind of a directed mutagenesis approach, very powerful. If you know the gene of interest and its sequence. So that's something to definitely keep an eye on. But we also have to take note of regulatory issues relating to gene editing. It seems quite accepted in US, but not necessarily in all other countries. So when we work on crops that are, for example, exported into many countries in the world, we have to be aware of perceptions and regulations. But certainly, it's a tool that we are exploring, at least at the bench level, at the research level, in ways to understand gene function, understand the relationship between gene sequences and the trait. So in those ways, it's a very interesting and powerful tool.
Yeah, so I would like to second this. I think that the latest and most important disruptive technology is gene editing. And if I can put it in a different, in a little different angle, basically on the backdrop of what I mentioned before, that the source of everything is genetic variation. It's not only a theoretical concept that eventually, with all the breeding that we're doing, we get to a point that will plateau, and the amount of genetic variation that is existing there is getting smaller. With these technologies, we actually no longer are dependent on natural variation. You can actually induce the variation in the direction that you are interested in, and it started with small changes of one or few nucleotides. And here at the Plant and Animal Genome Meeting, where I'm at right now, we heard all kind of developments which are extremely are very interesting, like swapping. Inducing translocations, swapping chromosomes arms. So I think there is much more that can be done and will be done with this technology. Another one, which is it's not exactly a technology, but it's more like a newer field in our space, is soil microbiome. We know how important is the environment for all the breeding effort, and here we're getting a new look, or we get a new understanding of the immediate environment of the plant or the root system by exploring the fauna that is existing in the soil around, or the soil that the plant is growing in.
Thank you, guys. This has been such an interesting conversation. I know myself I'm going to be going back through notes and stuff because I am not a super sciencey person, but I want to make sure I take in everything that you guys have said. So unfortunately, that is all the time we have today. But thank you to Beni, Ron, and Tom for being here. And for those watching, thank you for tuning in. And if you missed part of today's live stream, don't worry. We'll have it available on demand on Facebook, YouTube, and LinkedIn. And next week, please tune in as my colleague Mark Zinkowitz will have some guests on to talk about plant breeding targets. Thanks for tuning in and have a great day.